Hello everyone. Today we are going to deal with the topic plant growth and development. In this we are going to discuss about the concept of plant growth regulators, and we are taking up one phytohormone, that is auxin. So let's move on. When we talk about plants, we know that the plants undergo growth, differentiation and development. And these are the three very closely related events in the life of a plant. Now, when we talk about development, especially, we know that the sum of growth and differentiation both is considered as development, as not only cell division is important for the plant, but also the assigning functions to the newly formed cells. Therefore, it is said that sum of growth and differentiation is development. Now, when development is important for plant, we must know that what are the factors which affects development. So, when we talk about plants, there are two factors which are important, or we can say that it controls the development in the plant's life. These two factors are extrinsic factor, this one is extrinsic factor, as well as intrinsic factor. When we talk about extrinsic factor, extrinsic factors includes light, temperature, water, oxygen, nutrients, etc. But when we talk about intrinsic factors, they includes intracellular or genetic factors or intercellular factors such as hormones, and these hormones are also known as phytohormones or growth regulators. So here we are going to discuss about one of such intrinsic factor, one of those intrinsic factor which is known as growth regulator. So when we talk about growth regulators, they are also known as plant growth regulators. These plant growth regulators plays very important role. The other term for these plant growth regulators is phytohormones, where phyto means plants and hormones. It includes it can be natural or synthetic. So we can say that plant growth regulators includes plant hormones, but also includes non-nutrient chemicals, which is not found naturally in plants, that when applied to plants, it influences their growth as well as development. So when we talk about these plants, it is very important for us to understand what are these plant growth regulators. Now, plant growth regulators plays very important role, and these regulators can be categorized under 5 different categories. One is auxin, gibberellins, cytokinins, ethylines and abscisic acid. All 5 phytohormones play very important role in the development of plants. So today we are going to discuss about auxin, a very important phytohormone found in the plant. Now when we talk about auxin, let's discuss first about discovery and isolation. Now, Charles Darwin and his son Francis Darwin observed. So Charles Darwin and Francis Darwin observed that the coleoptiles of canary grass responded to unilateral illumination by growing towards the light source. So, Charles Darwin and Francis Darwin, they did experiment on canary grass coleoptiles. It responded to the light which when illuminated from one direction. That is why it is written here unilateral illumination was given to the plant, and it started bending towards the light source. And after a series of experiments, it was concluded that the tip of the coleoptile is the site of production of substance. That means that whatever was causing bending towards the light source was produced at the tip. That is the site of production of that substance, and it caused bending of coleoptiles. Later on, F.W. Wendt, he isolated auxin from the tips of Avena, and Avena sativa is also known as oat seedlings. So, from oat seedlings, coleoptiles of oats, F.W. Wendt isolated auxin for the first time. And it was found that auxins are of two important types. They can be categorized in two different categories. Number one, they can be natural auxins, and second, they can be synthetic auxins, that is made by man. So, auxin word is derived from a Greek word auxin, which means to grow, and it was for the very first time isolated from human urine. And this term auxin is applied to the indole 3 acetic acid as it promotes the growth. That is why it is applied, auxin is applied to the indole 3 acetic acid. In short, it is IAA. So, natural auxin is indole 3 acetic acid as well as indole butyric acid, that is IAA and IBA. But synthetic, which are made by man, they are NAA, naphthalene acetic acid, and 2,4-D. You must have heard about 2,4-D, 2,4-dichlorophenolic acid. So, this is synthetic auxin. So, let's talk about the most important experiments which led to the discovery of auxin, and it also gave us a clue that auxins are produced at the tip of plants, that means at the apical ends of the plant. So, it is very clearly shown here. You can see that when light is overhead, it is observed that auxin molecules are produced by apical meristem. This is apical meristem, this is apical meristem, and when sun is overhead, that means the tip is illuminated equally from both the sides. Then at that time, it is produced by apical meristem and distributed evenly at both the sides, But it was observed that if unilateral light is provided like this, then this unilateral light, when unilateral light is provided, then this changes. How it changes? When it was observed at that time that the shaded region, this is the shaded region, unilateral light from this side light was provided, and this is the shaded region. The chemical was produced at the tip and it started flowing or moving towards the shaded region. As a result, it causes bending of the shoot towards the light. And reason of bending of the shoot towards light is cell elongation. So because of cell elongation of this shaded region, this chemical was auxin. Auxin was produced at the tip. It started moving towards the shaded region, and which caused cell elongation, and hence we observe the coleoptile or the tip bending towards the light. I hope it is clear. Let's move on. Now, there are series of experiments which were performed by different scientists, which proved that auxin is produced at the tip. This is the site of production, and it is the one which causes cell elongation and bending towards the light source. So you can see here that Darwin, this is Darwin, he did one experiment where he took the canary grass, and this is the canary grass, and he cut the area, and when he cut the area, he found that this is a coleoptile, and top of the coleoptile, when removed, it showed no bending. There was no bending. But when coleoptile tip was present, it showed bending towards the light. And later on, he covered the tip. This is the tip region. He covered the tip region with the help of a cap. Again, there was no production of auxin, so it did not bend. So this was the first experiment where the chemical was not recognized. In the next experiment, it was found that the tip was removed. And a piece of gelatin was kept in between tip as well as the part. So this part is gelatin. So gelatin helped in, through gelatin, this auxin moved towards this side, so it showed bending towards the light. Now there was another experiment done by Paul where he showed that the tip was removed, cut, and the tip was placed on one side, and when he kept it on one side, auxin, when light source was provided, it was unilateral light, so auxin was produced and it started moving towards the shaded side, so it caused bending. In the last experiment, which was done by F.W. Wendt, he was the one who isolated auxin from the tips of coleoptiles of oat seedling. So it was found that when coleoptile tips were kept on agar blocks, then the tips were removed, and then this agar was cut into small blocks, and one block was kept on one side of the coleoptile. It was found that it showed bending, and the reason is that the agar block contained that chemical, that is auxin, which started moving towards the this side, and it showed bending of the coleoptile. So these are series of experiments which concluded that auxin is produced at the tip of plants, which causes bending towards the light source, and this phenomena is known as phototropism. I hope it is clear. And so let's move on. Now, when we talk about physiological effects of auxin, we know that auxin causes the most important effects, that is, it controls the growth of plant cells. So, it is clear that growth here means it is obvious due to cell division. So, it is observed that auxin also controls the cell division in vascular cambium, and it also causes differentiation of xylem. And it is the most important, this is the most important point which showed that the auxin promotes apical dominance. We will discuss about apical dominance. It also promotes, or we can say initiates, rooting in the stem cuttings. So you must have heard about tissue culture. In tissue culture, the callus starts, from the callus we see that new roots are produced, and this is because of the presence of auxin in the nutrient medium. So, in plants, natural auxin is present, which helps in initiating rooting in stem cuttings. It also promotes flowering in some plants like pineapples. It also helps in prevention of fruit and leaf drop at early stages. It means a plant, whenever a leaf falls or a fruit falls, before this, an abscission layer is formed between the petiole and the stem. This abscission layer blocks it gives all the nutrients first to the other parts, then it forms a blockage in that area, abscission, so that is known as abscission layer, so that leaf can fall off or fruit can fall off easily. But it should be prevented at early stages because leaf helps in photosynthesis, and fruit formation is also important for the formation of seed. So to prevent it at early stage, auxin plays very important role. It also promotes, but on the other hand, it promotes the abscission of older mature leaves and fruits. So it prevents fruit and leaf drop at early stage, but it helps in abscission of older mature leaves and fruits. It induces parthenocarpy. Parthenocarpy means seedless fruits and vegetables. So it helps in production of seedless tomatoes. 2,4-D it helps in killing dicots but not monocots. So 2,4-D and such auxin, a form of auxin acts as weedicide or herbicide. And the last one is it controls xylem differentiation and also helps in cell division. So these are the basic functions, roles performed by the auxin. Now let's move on to the next one. Now we are going to discuss about apical dominance and phototropism. These are very important phenomena. Now, when we talk about apical dominance, apical or terminal buds of many plants are very active, while it is found that lateral buds remain inactive. So, it is observed that if we remove apical buds, then the lateral branches grow very fast. And it is observed that this phenomena is known as apical dominance. Apical dominance takes place because there is higher concentration of auxin in apical buds than the lateral buds. So, because of this reason, the terminal buds grows faster than the lateral buds, and it is known as apical dominance. And if you will see gardener who makes hedges, gardeners always cuts the tips of the bushes so that lateral branches can grow very fast, and the that hedge becomes bushy. And in tea plantation also it is the same thing. They they uh, they cut only the tip region of the tea plants so that the lateral branches grow very fast and it becomes bushy. So this is phenomena is known as apical dominance. It is because of hormone auxin. On the other hand, phototropism is another phenomena in which plant bends towards unilateral light. That means light source from one direction. So plant shows bending towards it, and this is also due to flow of higher concentration of auxin towards the shaded side. So these are the two phenomena which are very important related to auxin. Here it is shown in this, it is shown that this is a plant. This is a plant where you can see abscission layer is formed. This is the abscission layer is formed, and this is abscission layer. Here you can see this is a stem and this is a petiole. So before falling off the plant, this forms a plant forms abscission layer here, and this abscission layer is formed so that this leaf can fall off easily. This leaf can fall off easily. So this abscission layer is formed, and here you can see that these are the lateral branches, and this one is the apical branch. So in apical branch, auxin is produced in large amount. That is why the growth of lateral branches does not take place. If we will take out this apical, if we will cut this apical area, it will promote the growth of lateral branches. So this is another picture which is shown where you can see that abscission layer is formed, and this is the petiole and this is the twig, and an abscission layer forms so that leaf can fall off. So this is a natural phenomena, but it prevents the early drop of plants. So application. Now let's come to the uses of auxin. Auxins are very important and they are used to initiate rooting in stem cuttings, as well as in it is used in nutrient medium in micro propagation, that is tissue culture. They are also used to induce parthenocarpic fruits, that means seedless fruit formation. And then these are also used into 4D is a synthetic auxin. It is used as a weedicide as it kills only dicot weeds, not the monocot weeds. So this is another application. It also promotes flowering in pineapples and lychee. And in some plants, it is used to prevent the premature fall of leaves, early fall of leaves, flowers, and fruits. So these are some applications of the auxin. Now, it is just a recap. Uh, auxin's growth. Auxin is growth promoting chemical. It promotes cell division and cell elongation, but always remember that it promotes cell division and cell elongation in the roots. The primary plant auxin found in plants, especially the natural auxin, is indole acetic acid, and synthetic forms of auxins are IBA and NAA, that is indole butyric acid and naphthalene acetic acid. And natural uses of auxins are: it is useful in phototropism, geotropism, apical dominance, and root formation. And we know that phototropism is growth of plants towards the light source, that means stem growth towards light source. Geotropism means plant root growth towards the gravitational forces, and apical dominance is a natural phenomena where terminal buds prevents the development of lateral buds on plant stem. So this is a natural phenomena by Auxin. I hope it is clear. And auxins are the most important growth promoters, and they play very important role in promoting the growth of plants through cell division. I hope you must have understood this much. Thank you.